So good morning, everyone. Yesterday we started our journey talking about gene editing from the scientific point of view. And during the day, we realized addressing the issues of life, responsibility, and governance, that science is not only an activity producing knowledge, but it is also a social practice, needing resources, needing spaces, needing sponsors, and so on, and also needing dialogue within the research groups. We saw a few examples at the end of each presentation, the people working together, and also within the scientific community in order to achieve a consensus between different competing hypotheses. So today we will start directly from the ethical point of view. We experienced yesterday that it's not easy to make bridges between disciplines, namely between, let's say, experimental knowledge and humanistic knowledge. We experience that when we talk about consciousness, for example, or about the meaning of replication in order to understand life. So we also experience that we cannot give it for granted. So today we will start directly from the ethical point of view, addressing the issue of organoids. And our speaker will be Professor Søren Holm. He is very well known in the bioethical circles. He holds a chair of bioethics at the Center of Social Ethics and Policy at the University of Manchester, Great Britain, and also at Oslo University, Norway. So, Professor Holmes, the floor is yours. Thank you very much.
And thank you very much for the invitation to talk about this now. Sort of everything I know about organoids, or at least about organoids recently, is because we've just finished a European project called Hybrida on the ethics and regulation of organoids. So that's sort of where all of this comes from. Not really.
Not yet. Okay, I will.
Okay, so this is what I'll briefly talk about. What is an organoid? Why do we create them? Then I'll talk about the range of ethical issues, general issues. Identify two types of contentious organoids. One we talked about yesterday already, neural organoids and embryo models. I'll focus the talk on those, and the focus on all of this is on human organoids. Now, human here is in quotation marks because the thing about human organoids, which is human, is that they're made from human cells. They might not be human in many other ways of taking that term. Now, what is an organoid? Well, it's a three-dimensional biological structure. We create them from one or more cell lines by directed growth and differentiation in the lab. And the basic function of an organoid is to be a model of a particular organ or a particular part of an organ. So you could have a liver organoid, which is supposed to model the liver. You can have kidney organoids. You can have cortical organoids, and essentially any tissue can be modeled in this way. But it's important that an organoid is not necessarily an organ. It is a model of an organ. And as with all other models, it could be the particular organoid could be a good model, or it could be a bad model, or it could be a good model of one function of an organ and a bad model of another function of an organ. So the fact that someone says, well, I have made a liver organoid, just tells us that that researcher has aimed at a model of the liver, doesn't say anything about how successful it is or what it is about the liver that they have aimed at doing. Well, why do we create organoids? Well, we create them for a whole range of different things. We create them to do basic research. We create them to do pharmacological and toxicological testing. And they are part of the sort of broader vision of replacing animal models. We can create them to produce biological products because by producing things in human cells, you can get the right glycosylation of your proteins, which is difficult to do in other ways. They're also part of the precision medicine vision. So the idea is that if you have a particular disease, a particular cancer, we can grow a tumor organoid and then we can test all of the different drugs on that and find the drug combination which is uniquely suited for you. And then there's, oops, wrong button. There's also a vision of therapeutic use in the future. And some people project the vision that eventually what we'll be doing is not growing organoids, we'll be growing organs, and then we'll use them for transplantation. So it's a field where there are lots of visions, and there's also a lot of reality, but the visions and the reality do not match yet. Okay. There are a range of general issues about organoids which are essentially related to the fact that we make them from cell lines. And there are loads of ethical issues about cell lines which we have been discussing for a long time. Obviously, it might matter what kind of cells we use. Are we using human embryonic stem cells, which raises a whole range of ethical issues on their own, or are we using, for instance, induced pluripotent stem cells based on cells taken from adults, which doesn't raise the same issues as the human embryonic stem cells. Then there are issues about the initial donation. Is that the right type of consent? To what extent do people have a right to withdraw when their cells have been turned into a cell line? Even if they can't withdraw, should they have some kind of control? Should they be able to say, yeah, university researchers can use my cells, but not the evil tobacco industry, and so on. And then there's an issue about, well, if, for instance, a particular cell line becomes important for some kind of commercial product, well, who should share in the financial benefits from that? But none of these are specific to organoids. They are cell line problems. There's also a problem about information that obviously from a cell line we can derive genetic information, which is genetic information about the original donor. And in the European context, that creates enormous problems because it means that a lot of users of cell lines activates the general data protection regulation and can therefore make work for many lawyers for many years. But again, this is not an organoid problem, this is a cell line problem. There are, however, two kinds of organoids which are generally accepted to be in some way contentious. And the first one is neural organoids. And I will let the researchers here off and say they were badly advised by the Cambridge University press office, because what they're doing is not growing brains. They're growing neural organoids. And the main concern about these, which we already raised yesterday, could these organoids become sentient or conscious? And that concern is partly fueled by people calling them things like mini brains. Okay, so this is the concern. Well, what is a neural organoid? Well, it is a 3D model of parts of the central nervous system. Again, we grow them through directed differentiation of stem cells. We can also, if we grow organoids of two different parts of the central nervous system and then co-cultivate them, we can get them to the neurons in them to interconnect, so we can get what's called assembloids of two different ones. One thing which is important is that currently these are non-vascularized. They do not have arteries or veins or blood circulation or any other circulation. So their size is restricted by the maximal diffusion limit for oxygen. So if they grow too big, the sensor dies. Now, of course, you can do things. You can increase the oxygen concentration. You can increase the pressure. So you can grow them slightly bigger. But there is still a size limitation on the way we currently do these. That's quite important. So we have two questions. Can neural organoids develop sentience or consciousness? And how should we think about and regulate neural organoids in light of the answers to this first question? And the answer to the first question is probably yes. Or if it's not yes, at least we can't decisively say that it is no. It could be the case that if we could grow a sufficiently complex neural organoid, it would develop the ability to feel something, or maybe the ability to know that it feels something, which is a higher order ability than just having the feeling. However, I will try to convince you that at the moment, the answer for any organoid we can grow at the moment and in the foreseeable future is actually no. That although it's a conceptual possibility for very complex neural organoids, it is not a possibility for the kind of organoid we can do now. First of all, current neural organoids and assembloids are small and relatively simple structure compared to even the simplest mammalian brain. Now, I couldn't find a picture of a slice of a brain of a naked mole rat. So here you have a normal mouse. But I hope that you can see that even a normal mouse brain is quite structurally complicated. We have the cortex, we have the hippocampi, we have other structures here, and so on. And even the smallest mammalian brain, which is apparently the one of the naked mole rat, at least among those that anyone has ever counted, has a lot of neurons, many more than any organoid we can currently grow. The second observation, which might be important to make, is that computational ability is not sentience or consciousness. So a couple of years ago, we had this, that in a two-dimensional neural network, you can teach the two-dimensional neural network to play Pong, this video game where you have to sort of move a bat and then reflect the ball. Now, I'm old enough to actually have played Pong on a Sinclair ZX81. And it's not a very complicated game. Now, first of all, one important thing which got lost in the press is that the researchers did this with both human cells and cells from mice. And both types of cells can actually play Pong. The human cells outperform the mice cells with nuances on average, but both of them can play. But the mere fact that something can play Pong doesn't show that it is conscious or sentient. I'm fairly certain that my ZX81 computer, with all of its glorious 64 kilobyte extra memory which I bought for it, was neither sentient nor conscious, even though it could play things that were more complicated than Pong. And my guess is that none of you think that your current mobile phone is conscious. And of course, your current mobile phone is much, what it can do is much more complicated than this. So the mere fact that we have computational ability doesn't show that we have sentience or consciousness. I don't expect you to read this. I won't read it to you either. The main point is that there are a number of competing theories of how the brain generates consciousness. All of them have some empirical evidence, but we are far from actually knowing which of these theories is the right one. None have sort of really compelling evidence that it is the theory of how this is happening. And this is why it's still a hard problem. We don't really know how a very complex neural organ does this. So there are problems sort of at the theoretical level as well. And finally, or maybe not finally, almost finally, this is where the term human becomes really important, because there is a large risk of equivocation on that term. Now, the fact that a brain organoid is human, even if it had consciousness, does not mean that it will have a human consciousness phenomenologically. It will not feel and think like you and I do. And there are at least 3 reasons for that. First of all, because it's unlikely to structurally replicate our brain. Secondly, as has been pointed out by a long time ago, the French philosopher Maurice Merleau-Ponty, more recently by Antonio Damasio, embodiment matters quite a lot. We are not a brain that have a body. We are a brain in a body, and the way our brain functions and the way we think is dependent on our very specific embodiment. If we were embodied differently, we are likely to think differently. And brain organoids are going to have a different embodiment, a very different embodiment than one we have, even if they are in a robot. And finally, it will have developed its consciousness in a different life world. So we get the problem which already Ludwig Wittgenstein foresaw when he spoke or wrote about a conscious lion. If a lion could speak, we could not understand him. Why couldn't we understand the lion? Well, mostly for Wittgenstein, because the lion lives in a completely different life world than we do. So, whatever kind of consciousness a neural organoid is likely to have, it is not the kind of consciousness that we have. It is not going to think human thoughts, even though it is biologically human. It's going to think organoid thoughts about growing up in a lab, in a petri dish, and so on. The final observation is, of course, that in one sense, maybe there is no problem, because we are already fairly familiar with interacting with sentient and conscious neural organs. They just happen to be embodied in animals. So this worry, well, maybe my organoid can feel pain. Yeah, well, maybe your organoid can feel pain, but so can your mice. And so can the octopus and the other cephalopods that, at least in some countries, we now protect. So we are fairly common with regulation. Whether we get this right is another question, but we are fairly common with regulation of sentient and potentially conscious neural organs. At the moment, they just happen to be in animals. So my regulatory conclusion is that, yes, there are very interesting philosophical issues about sentience and consciousness in human organoids, and I'm happy to write long philosophical papers about these very interesting issues. But there's no need for current regulatory intervention, because the ones we can do now and in the foreseeable future are definitely not conscious or sentient. And if and when we really start worrying about, yeah, these are now getting so complicated that they could be feeling pain or pleasure or whatever it is that we think they might feel, the best analogy to draw on is not human research ethics, but animal experimentation ethics. Because these are not going to be initially anything like humans, they are going to be alien in the very real sense of something being other. Okay, the next type of contentious organoids is embryo models, because from stem cell lines we can make models of embryos. The problem is that we have scientific development, we have uncertainties, and we have ethical disagreements. Now, about the neural organoids, we don't if a neural organoid really had the kind of consciousness that we have, we wouldn't have much disagreement about what we should do and shouldn't do. But here we also have ethical disagreement. Now, this is not a new problem. Scientists bring about new ways of creating embryos all the time, and to the delight of ethicists like me, we can then roll out arguments that we have made before and recycle, and it makes writing the papers much easier. Now, the uncertainties are partly ontological. What are these things that we're creating? They're epistemic. What can they do in the future? They're ethical. So we have a regulation under uncertainty problem. So essentially, the issue arises because organoid technology allows us to create entities that model early embryonic development. So if you can take 3 different types of cells, for instance, mix them, if you grow them in the right way and direct them in the right way, you will then get something which is a model of an embryo. This is further, we are better at doing this in mice than we are in humans, but this is a very fast-moving research field, and people can do things that get closer and closer to being embryos and getting closer and closer to later embryonic stages. And this is what raises the ethical and regulatory issues. How should we conceptualize these models, and how should we regulate them? Now, one way of doing this is to ask whether they are embryos. So, sort of trying to decide of their sort of ontological status, are these things that we are producing embryos? Now, one first thing to note is, of course, that it relies on the implicit assumption that if we know that whether they're embryos or not, we also know what to do with them. But as I pointed out, we have ethical disagreement as well. So that assumption can't necessarily be taken for granted. There is no philosophical, societal, political consensus about how we should actually treat embryos. And there's probably no hope, even if we dialogue for years, which we have actually done in this field, that we are going to reach consensus. On the other hand, in many countries, the lack of consensus hasn't meant that we haven't actually regulated embryos. So we find political compromises even though we don't have the consensus. Now, if we try to solve this problem by saying, is this an embryo? Well, then we need to know what an embryo is. And that has quite a lot of different possible answers. A developmental stage in the development from a fertilized egg to a newborn, a human being, a possibly ensouled human being, an entity with active potential to become a human being, and so on. And it's very likely, unlikely that we can reach consensus. And the question, is X an embryo, will have a different answer according to which of these we take. So if we, for instance, take the first one, a developmental stage in the development from fertilized egg to newborn, none of these things could ever be embryos because they are developed in some other way. They are definitely not coming from a fertilized egg. And then we have the problem. So when does a model become the real thing? When does homology become identity? And given that I'm originally Danish, I've illustrated this with Lego models, and I hope you'll go and buy lots of Lego. Now, none of these are cars. They are models of cars. Now, when does a model of a car really become a car? And as I said previously, some of the answers to the ontological question entail that the cell-based models we discussed could never be identical. We also have a problem about the human embryo models, that some of the things we would do eventually in animals, we will never do in human beings for ethical reasons. Now, in animals, we could implant, if we really thought, well, this is now really so close to an embryo that it is sort of could develop to term, we could implant it into the uterus of that animal and see, well, does it develop to term? Now, we are obviously not going to do that with human beings. We are not going to sort of grow embryo models in this way in the lab and then implant it into a human and see a woman and see what happens. Sort of no ethical committee in the world would approve that. And that is sort of, and that means that in humans, sort of the final proof of biological identity between the model and an embryo will never be reached, because that is going to be complete development from the model to an adult of that species. Then there's a question about, well, we have the same problem here. Sort of these models are currently not vascularized. They're not, sort of, we don't have anything on which to implant them so that they could develop blood vessels. So they can only develop to the limit for oxygen diffusion as well. But people are working on this, obviously, as scientists do. So when we think about these, sort of, should we think about what we can do now, in which case, well, none of them are going to develop any further than a certain size, or should we think, well, these might, this might be solvable in the future. And to what degree should we take already existing possibilities into account? And I'll come back to the later point. Then, of course, we have sort of regulatory uncertainty as well. What is the proper objective or aim of regulation? Should we regulate sort of on precaution or opportunity? How should we think about sort of the distal harm and distal benefits of this? Obviously, sort of what's driving this is the promise that there are important benefits from this line of research in the future. It will help us to understand early embryonic development, which would help us to improve IVF treatment or help us to prevent various forms of disease and so on. Are we looking for a compromise solution? So are we looking for consensus, or are we looking for a compromise solution? Now, there are regulatory suggestions, and I'll just talk about two of them and try to show that they share a common problem. The first is the International Society for Stem Cell Research Guidelines 2021. I should say they're currently on the revision, so we don't know what the new ones are going to be. And they make a distinction between non-integrated stem cell-based embryo models and integrated stem cell-based embryo models, which I'll come back to. And then there is another suggestion from a prominent researcher in the field and co-authors that we should redefine embryo to include environment. So we have an extended embryo, and then we have a legal definition that an embryo is a group of human cells supported by external elements fulfilling extraembryonic and uterine functions that combined have the potential to develop into a fetus. And then Rivron has a testing for what he calls tipping points. And I'm just going to talk about Rivron, even though it is Rivron and other people, including people who are more knowledgeable about ethics. Now, the ICEEL guidelines make these distinctions. So on the one hand, you have the non-integrated models, which experimentally recapitulate some, but not all aspects of the peri-implantation embryo. For example, they might lack differentiation of the embryonic sac, or they might lack the embryonic disc. So these stem cell-based models, because they're not complete, do not have any reasonable expectations of specifying additional cell types that would resolve information of an integrated embryo model. So essentially, these have features which means that we can be certain that they would never be able to develop into a fetus. there are things that they lack, which a normal embryo has, which means that they can't develop into a fetus. Whereas the integrated stem cell-based embryo models are those that contain the relevant embryonic and extra-embryonic structures and could potentially achieve the complexity where they might realistically manifest the ability to undergo further integrated development if cultured for additional time in vitro. And then the guidelines also make the valid point that the previous restrictions, so for instance, the 14-day or primitive streak rules, were not written to apply to these, and it's difficult to apply them because there are no 14 days here from that you can measure and so on. So essentially here it's between sort of those models we know could never develop further than a certain stage because they're deficient in some way, and those that can. And the ICEEL then has suggestions for a tiered way of looking at the ethics, where we should look more carefully at the ethics of the integrated ones. Now, Rivron says, well, there's a biological definition of the embryo, but we should move, in this context, we should move away from it to a legal definition. So the embryo are the human cells with active potential to form a fetus, the support elements fulfilling extra-embryonic functions, and the support elements fulfilling uterine functions. So it's sort of the whole collective of these. And then they suggest two tipping points. One of them is essentially sort of a morphological one. So does this thing look like the human thing that develops? And depending on where you are, in which jurisdiction you are, you can say, well, yeah, we are going to put the cutoff point here, or we're going to put the cutoff point here. So it's a question about, in one sense, about morphology. Of course, they are also making the point that there are other ways to analyze these embryos and the cells they contain, sort of to say something more precise about them than this. And then they have their other test, which is that what we should look at is we take our integrated embryo model, which we have developed in an animal, we implant it, so if it implants in mice, it is sort of at a certain level. Well, if we can make a model in pigs, we should perhaps think that we're closer to humans. If we can make one in monkeys or maybe even in primates, then we're even closer to humans. And again, each jurisdiction sort of has a slider down here and reaches a decision about where it is. Now, both of these rely on essentially an assessment of the potential of the model. And this is, of course, again, something which people like me think is a great thing, because, well, I wrote my master's thesis on the argument for potential in embryos in 1990, which is a long time ago. So it's quite interesting to see potential coming back in this way. But this creates a problem, because how do we determine the functional potential of human embryo models for regulatory purposes? Because they are technobiological artifacts. Now, we don't know the potential of an individual normal human embryo. But we do know the potential of the type. So we don't know the token potential, but we do know the type potential. But this is a question of, we don't know the type potential of any of these technobiological artifacts. They are made de novo, they are maybe the iteration of something, but what we're asking is, sort of the last development, what is the potential of that? And there is a problem that model potential is malleable. That the inherent functional potential of a human embryo model can be changed by technical intervention. Now, very obviously, we can reduce the potential by removing things from it. More importantly, it's also plausible that we can increase potential by technical developments. Now, we haven't done this in embryo models, but until very recently, it was not possible to clone rhesus monkeys. The clones that you had all had problems in developing a normal placenta and therefore did not go to term. But Liao and so essentially sort of the cloned rhesus monkey was insufficient, but Liao and colleagues came up with an ingenious solution, because if you have a normal rhesus monkey blastocyst, you can take out the inner cell mass, you can then put in the inner cell mass from your clone, and then, hey presto, it goes to term. Well, not all of them, but it is possible to. So what they have done is to change the potential of a particular kind of embryo-like thing, a cloned embryo, to something more. It now has potential to go to term and turn into a rhesus monkey. Again, this is not a new thing. It occurred as well when someone suggested altered nuclear transfer in relation to the use of stem cells. So we have the problem that potential is not stable, it can be changed, and then we have a specific problem for Riveron suggesting that the tipping point in related species depends on an implicit assumption that either the human development, the development of human embryo models always lags behind the development of animal embryo models, or that for the same level of development, a human embryo model will always have less function or potential than an animal embryo model. And that is the two assumptions you need. You need one of them to be true to get a valid conclusion that the tipping point will always be reached before the human embryo models have that function. So the problem is that we don't have any reason to believe that these assumptions are analytically true. They might be true, but if they're true, it's an empirical question. So they're contingently true. So Riveron's second tipping point actually doesn't necessarily work. It doesn't work as an ethical marker of the potential of human embryo models. Can the regulatory questions be resolved? Well, probably. Now, we are not going to resolve the underlying fundamental ethical uncertainty. So what we are looking for is probably some kind of compromise solution. Now, our problem is that we can't, this is one of the questions we can't really leave unsolved. Researchers need an embryo definition. They need to know what they're doing, what the legal status of the human embryo models they are developing are. And then, of course, we also have regulatory problems about, so should this be regulated nationally or internationally? Should it be by law? Should it be by self-regulation? Would it be by something in the middle? And these are the references. Thank you.
Thank you very much, Professor, for this presentation. It's very complex, many issues involved. We can maybe postpone a little bit the coffee break since we just had breakfast a few minutes ago. So, and give some more place to the discussion. So there, I see already a few hands raised.
Thanks very much, Søren. I think this is more of a clarificatory question, but I didn't quite catch on to what you were getting at the end with the different potentialities, the slide with the 2 Turing tests. So I just wanted to clarify, is what you're saying that there are different kind of probabilities of the potentiality between the potential human embryo models and the animal direct animal reproduction? Or is it not the probability of leading to the end potential, but the value of the end potential? Because is that what you were getting at with the problem with well, this would count out animal experimentation?
No. The problem with the second of Riron's tipping point suggestion is that the suggestion is that as a society we can say, at the point where this can be done in mice and bring about a healthy mouse, we can say, well, that's the point where we want to stop, because if we stop at that point, the human embryo models who are at the same level of development will definitely not be able to turn into humans, even though we're never going to be able to say, because we can't do the experiment, but at that point we can be certain that they can't do it. Now, if you can do it in a pig, we can be slightly less, but you could say, well, that's the degree of certainty, or the degree of certainty is the monkey or the primate. And the problem with that is that it does rely on the fact that we either have to believe that there's something biological about human embryo models, which means, or we have to believe that they are always lagging behind in development. So if we stop development at a certain point, they won't have exactly the same potential that, let's say, the mouse model has. And both of those assumptions are essentially empirical and not conceptual.
Thank you for your comprehensive presentation. I'm Maurizio Muraca from Padua. I also appreciated the fact that you took the responsibility to give some answers. Although I don't fully agree with some of your answers. You stated that we can assume that there is no consciousness in these neural organoids for a series of reasons. But yesterday we all agreed that we do not have a definition of consciousness. And probably we should consider this limitation. Regarding embryo organoids, you insisted on the potential to develop. However, there is another question behind. When does a human being start existing? For instance, probably this is not the right example, but there are several embryos that naturally do not stop evolving and they are eliminated, also at very early stages. And because of this, they are not human beings.
Well, as I said, I have personal views on the second question, but I know that my personal views are not shared by everyone. And I think in this particular, if you think that very early, let's say, zygotes are human beings with full or nearly full moral status, which I think is a possible position, you still have the problem that these models are not zygotes. They've never been zygotes. They are not. They come about in a completely different way. So the type of arguments that sustain the idea that the zygote is a human being may not apply to these. And I think that is why all of the regulatory suggestions go to potential. And I think, well, I haven't seen any, at least to me, convincing way of solving the problem of saying exactly when the model becomes the real thing. And that's the problem you would have to solve. Now, in the first thing, yes, of course, there are theories of consciousness which means that there is consciousness in these. If you accept panpsychism, then sunflowers are conscious, undoubtedly. So yes, we can't. I can't. There's no argument which would convince a panpsychist that these things are not conscious. And I'm not trying to provide that argument. The argument I'm aimed at is towards the people who accept that consciousness is in some way produced by the human brain. It might be produced by other brains as well, but it is produced by something which has a very significant degree of complexity of a specific kind. And my point is just that the current neural organoids we can make do not have that along a whole range of different things. And even if they had, what they would have would not be human consciousness. They would not think thoughts like we do. If they thought thoughts, they would be other kinds of thoughts.
Thank you very much for your presentation and for your thoroughness in working through these things. I'm intrigued by this use you have of sort of it's not quite the same, right? And then using that to distinguish between how we would treat something, maybe if not ontologically, but morally, all right? It's a fascinating argument because there are people today who will say, you cannot tell me what to do because you didn't live my life. And so if we take your sort of logic and extend it, it leads us to a society where no one can regulate anybody else, right? Because you can't be in my brain and you don't understand me. So going back to Wittgenstein, how do we know if lions could speak that we couldn't understand them? That's a presumption we are making, right? That they don't think the way we think. So it seems to me it depends on which direction you seem to be coming from. If you're coming from the direction that says we have to establish the identity or an identical level of development in terms of context as well as in terms of degree, then you can push to a level that it may be farther along what we might call a progressive scale. If you come the other direction and say, we must presume, or we should presume, that things might be similar enough that there would be a relationality. So for instance, you could argue that 3 girls made by Dr. Huh did not develop the same as anybody else, which is true. They didn't develop the same as anybody else because no one else used CRISPR on a human embryo. Therefore, are they animal models? Are they humans? So depending upon the degree of which the intervention occurs, right, where are you going to draw the line? And I think that might come from the sort of presumptions that you make in terms of what you're trying to achieve, which is either protection or, you know, advancement of some kind. So that I'm just curious as how you come out, you know, in that kind of balance. Thank you.
Thank you, Kevin. Maybe if we keep synthetic our Q&A, we try to have 2 more questions.
Okay. Well, first of all, I want to make clear that I am not particularly sympathetic to standpoint epistemology. So sort of the claim that sort of you can't understand anything because you're not me, I think is just plainly false. But that's not the claim I'm making. It's not about what the thing thinks about itself. In the embryo case, I don't think sort of precaution or in whatever form you take solves your problem, because embryo models differ from embryos on a whole range of scales. So unless you want to sort of be hyper cautious and say all embryo models are embryos or should be treated as embryos, then you do get the problem that you have to decide where to apply your precaution. And that then becomes sort of a question of deciding, well, what is important about these things? Is it their morphology? Is it what they could do? Is it what they could turn into? So I think you do have a problem, even if you sort of wanted to say, well, can we be certain that they don't have whatever it is? Can we be certain that they are not ensouled? And my answer is probably for some of them, yes. And for some of them, depending on sort of your particular account of when and why ensoulment happens, you probably wouldn't be as certain, but again, you would need criteria.
Very briefly, there are a lot of philosophical uncertainties which you have brought up very interestingly. But there's one thing that doesn't seem to be uncertain at all, and that is the fact that you have defined organoids as models. So what exactly is the ontological status of a model? And in a way, that seems to be the starting point of the potential uncertainty, right? Is it a kind of postulatory definition? or is a model in a way assumed as a, shall we say, counterfeit double of the real thing? But then you have the question of how is it that the counterfeit double all of a sudden poses a series of real problems. So if it poses a series of real problems, it wasn't really a counterfeit double to begin with. So I don't know if I make myself clear, but I'm struck by the fact that the ontological status of the model is taken for granted. Whereas for me, that sort of postulatory starting point is the beginning of all the philosophical questions.
Okay. Well, most models are not counterfeits in any sense, in that they are not false in any sense. But of course, models are many things. Some are purely structural models, some are functional models, some are a combination of those two. And some models are deliberately partial, that they're not intended to model everything. They're only intended to model something of the function, for instance. But I take it to be sort of relatively certain that the Lego model of the Saturn V rocket that my son has in his former bedroom is not the Saturn V rocket. It doesn't have the same size, it's not made of the same materials, it cannot do the same things. But nevertheless, it is a model, and it is a real model, and it's a good model for what it is. A Lego model of a Saturn V rocket. So I think that for any model you can raise a number of questions about how similar it is to the thing it models and what the importance of these similarities are. But that doesn't make most models, and definitely not models that we make in the lab, false in any way. But of course, it's a large problem. I started out before I became allergic to rats a long time ago as an experimental neurophysiologist, and we were developing disease models of various diseases. And of course, what you do in a rat, in many cases, is very far from the thing that creates the same disease in a human being. But it's nevertheless a model, and you can then talk about exactly where the homologies and differences are.
Thank you very much. So I think we have to stop here. We will have the coffee break. We will continue the discussion during the coffee break. At least we had, thank you very much for your presentation. And to all the participants, because we at least had a taste of how to do a dialogue between different perspectives about the same phenomenon. Thank you very much.